Every living cell contains a substance described as genetic material. This material is composed of nucleic acid, DNA. It has an underlying linear structure, possessing segments called genes, the products of which direct the metabolic activities of cells. An organism's DNA, with its arrays of genes, is organized into structures called chromosomes, which serve as vehicles for transmitting genetic information. The way chromosomes are transmitted from one generation of cells to the next and from organisms to their descendants must be exceedingly precise. It is not possible without cell division. Two major processes involved in the genetic continuity of nucleated cells are mitosis and meiosis. Although the mechanisms of the two processes are similar in many ways, the outcomes are quite different. Mitosis leads to the production of two cells, each with the same number of chromosomes as the parent cell. In contrast, meiosis reduces the genetic content and the number of chromosomes by precisely half. This reduction is essential if sexual reproduction is to occur without doubling the amount of genetic material in each new generation. Meiosis is part of a particular type of cell division that leads to the production of sex cells, gametes or spores. We will discuss it in a separate video. The focus of our video today is the process of mitosis. As you can see here, there are steps involved before the cells divide. We will talk about them in detail. Before going through the different stages of mitosis, it is wise to know about the structures known as chromosomes. These structures carry all the genetic information in a cell. Let me explain a bit more. The word chromosome means colored body, referring to the capacity of these structures to take up certain histological stains more effectively than other cell structures. Each chromosome is composed of an extremely long molecule of DNA complexed with proteins and RNA to form a substance known as chromatin. They disperse throughout the nucleus during the interphase of the cell cycle A, when the cell is not dividing but becomes compacted during mitosis and meiosis. A typical human nucleus has a total of 46 chromosomes, which makes up the 23 pairs of chromosomes. These cells are known as diploid cells. There are also the haploid cells that have only half the chromosomes. Those are sex cells that are produced after undergoing meiosis. Strictly speaking, mitosis is that portion of the cell cycle during which the hereditary components are equally partitioned into daughter cells, and the genetic material passed from the parent cell to the daughter cells is identical. The process of mitosis comprises stages of prophase, metaphase, anaphase, and telophase. Normally, cells linger in a stage known as the interphase before they undergo mitosis. This means they are in between the state of mitosis. During interphase, the long strings of DNA are loosely coiled together. That mess of DNA is called chromatin. But as the mitosis process starts, lots of things start happening in the cell to get ready for the big division. One of the more important things that happen is that this little set of protein cylinders next to the nucleus, called the centrosome, duplicates itself. These centrosomes regulate other organelles of the cell during replication and maintain the structure of the cell as well. Simultaneously, the DNA starts to replicate itself under the action of different enzymes. After the arrangement of centrosome and DNA, replication, the cell enters the first stage of mitosis called the prophase. The chromatin condenses on itself and coils up to produce thick strands of DNA. This condensed form of DNA, wrapped up in the proteins, is known as chromosomes. And at the same time, the centrosomes align themselves along the sides of the nucleus. Now, while the chromosomes are forming, The nuclear envelope gets out of the way by completely disintegrating. And the centrosomes then peel away from the nucleus and start heading to opposite ends of the cell. It becomes apparent near the end of prophase that each chromosome is a double structure split longitudinally, except at a single point of constriction, the centromere. The two parts of each chromosome are called sister chromatids because the DNA contained in each of them is genetically identical, having formed from a single replicative event. As they go, they leave behind a wide trail of protein ropes called microtubules, running from one centrosome to the other. As you may remember from our discussion of the cells and their organelles, microtubules aid in giving the cell a certain amount of structure, and this is precisely what they are doing in this instance. After this, the next phase in mitosis begins, which is the metaphase. Some scientists also divide it into protophase and metaphase. In these phases, the migration of chromosomes occurs, and it's the longest phase of mitosis. It can take up to 20 minutes. The distinguishing event of the two ensuing stages is the migration of every chromosome, led by its centromeric region, to the equatorial plane. The equatorial plane is also referred to as the metaphase plate. In some descriptions, the term prometaphase refers to the period of chromosome movement, and the term metaphase is applied strictly to the chromosome configuration following migration. Migration is made possible by the binding of spindle fibers to the chromosome's kinetochore, an assembly of multi-layered plates of proteins associated with the centromere. This structure forms on opposite sides of each paired centromere, in intimate association with the two sister chromatids. Once properly attached to the spindle fibers, cohesin is degraded by an enzyme, appropriately named separase, and the sister chromatid arms disjoin, except at the centromere region. A unique protein family called sqadia protects cohesin from being degraded by separase at the centromeric regions. At the completion of metaphase, each centromere is aligned at the metaphase plate with the chromosome arms extending outward in a random array. The events critical to chromosome distribution during mitosis occur during anaphase, the shortest stage of mitosis. During this phase, sister chromatids of each chromosome, held together only at their centromere regions, disjoin from one another, an event described as disjunction, and are pulled to opposite ends of the cell. For complete disjunction to occur, sqadia must be degraded, reversing its protective role. The cohesin complex, holding the centromere region of each sister chromosome, is then cleaved by separate. And sister chromatids of each chromosome are pulled toward the opposite poles of the cell. As these events proceed, each migrating chromatid is now referred to as a daughter chromosome. The steps that occur during anaphase are critical in providing each subsequent daughter cell with an identical set of chromosomes. In human cells, there would now be 46 chromosomes at each pole, one from each original sister pair. Lastly, we have the telophase. Telophase is the final stage of mitosis. At its beginning, two complete sets of chromosomes are present, one set at each pole. The most significant event of this stage is cytokinesis, the division or partitioning of the cytoplasm. Cytokinesis is essential if two new cells are to be produced from one cell. Other events necessary for the transition from mitosis to interphase are initiated during late telophase. They generally constitute a reversal of events that occurred during prophase. In each new cell, the chromosomes begin to uncoil and become diffuse chromatin once again. While the nuclear envelope reforms around them, the spindle fibers disappear, and the nucleolus gradually reforms and becomes visible in the nucleus during early interphase. At the completion of telophase, the cell enters interphase. With this, here the mitotic division of cell comes to an end and the cycle is complete. Thus, two cells are produced at the end of mitosis that enter the state of interphase. Check out our video on the process of meiosis to learn about the cell division of sex cells.
Watch our medical videos anytime and anywhere. Download sqadia.com app now.